Imagine unlocking the genetic secrets of ancient Egypt with technology that wasn't even possible just a generation ago. That's exactly what scientists accomplished when they successfully reconstructed the genome of an Egyptian man who lived approximately 4,500 years in the past. This breakthrough isn't just a scientific triumph. It's stirring up conversations about who the ancient Egyptians truly were and how they connected with neighboring cultures. But here's where it gets controversial. Does one person's DNA really tell the story of a whole civilization? The team, led by Adeline Merez-Jacobs, managed to extract and sequence this invaluable genetic material from remains found in Egypt, a region infamous for its harsh climate and challenging conditions that are typically hostile to DNA preservation. Given the heat, sand, and centuries of environmental changes, many experts believed it was nearly impossible to recover usable genetic data from that era. So, how did they pull it off? Let's break it down for beginners. Genome sequencing is the detailed reading of an organism's entire DNA code, which in humans is made up of around 3 billion chemical bases labeled A, C, T, and G. Think of it as reading the world's longest biological instruction manual. Sequencing technology has come a long way since the late 1970s, culminating in the landmark completion of the human genome in 2003. However, working with ancient specimens took things to a whole new level of complexity, namely because time, temperature, and microbes break DNA down into tiny, scattered pieces. Initially, methods like growing DNA in bacteria were time-consuming and often produced limited results. The real game changers were the development of PCR, polymerase chain reaction, in the 1990s, which made millions of DNA copies in mere hours, and next-generation sequencing machines that arrived in the mid-2000s, capable of reading countless DNA fragments simultaneously. These innovations didn't just make ancient DNA analysis faster, they made it far more accurate. Thanks to these advances, over 10,000 prehistoric genomes have now been pieced together worldwide, from Neanderthals to ancient farmers. Still, extracting viable DNA from tropical regions, like Egypt, remains exceptionally difficult because heat and moisture rapidly destroy genetic material. That's why this latest feat is so remarkable. So, what set this particular project apart? For starters, the scientists retrieved a fairly high percentage, about 4 to 5%, of genuine human DNA from the remains, which is quite rare in such environments. To put that in perspective, samples from living humans typically yield 40 to 90% human DNA, whereas ancient samples from hot regions often contain little to none. The secret? The man was entombed in a ceramic vessel within a rock-cut tomb, a unique burial practice that shielded him from the ravages of time and environmental extremes. Even so, the majority of the genetic material came from bacteria and soil organisms, rather than the individual himself. To maximize what little was available, researchers filtered out the most damaged fragments and used cutting-edge sequencing equipment at the Francis Crick Institute to generate close to 8 billion DNA reads. The results allowed the team to virtually reconstruct the genetic blueprint of the so-called Nuerat individual, making his DNA the oldest ever decoded from Egypt. Does this mean we can decode more ancient genomes from Egypt? That's the hope. This study didn't rely on brand new technology, but rather on an innovative combination of the best tools and methods in existence. By refining and optimizing these approaches, scientists proved that meaningful genetic data can sometimes be retrieved from areas previously thought hopeless, potentially revolutionizing our understanding of Egypt's ancient peoples. Now, let's get to the big discovery. What did the DNA actually reveal? By comparing the genetic data from this individual to those from ancient populations in Africa, Western Asia, and Europe, scientists found that about 80% of his ancestry tracked back to earlier North African populations. The remaining 20% pointed to the Fertile Crescent, mainly ancient Mesopotamia, a region now split among Iraq, Syria, Turkey, and Iran. In straightforward terms, This genetic evidence confirms long-suspected cultural and biological exchanges between Egypt and its eastern neighbors, reflecting the movement of people, animals, crops, and even writing systems like cuneiform and hieroglyphs across vast distances. And this is the part most people miss. Until now, no Old Kingdom Egyptian had been genetically sequenced, so these findings don't just echo earlier archaeological theories. They offer concrete, DNA-based proof. Of course, it's important to stress that one genome can't capture the full diversity of ancient Egypt, a civilization known for its complexity and multicultural layers. But this study opens the door for more discoveries and greater debates. Here's a final thought sure to spark discussion. Will future genetic studies rewrite what we think we know about the origins of the ancient Egyptians? Or could focusing too much on DNA distract from the rich cultural tapestry revealed by archaeology and historical records? Do you agree with the idea that one individual's genome can reshape our historical narratives? Or do you see this as just a tantalizing first step? Share your opinions and let the debate begin.